So, so welcome to our next session about in vitro models. But before we start, Ricardo asked me to announce that the online participants should raise their hands on the screen after the seminars when they want to discuss with us. So that's very important to see if somebody is interested. So, but now let's start with the first seminar. It's Arpna Sharma, and she will give us some insights in isolation and characterization of bovine luteal cells.
So, good morning to everyone, and very happy to see you all. And so in this...
Microphone?
Yeah? Yeah, okay. So, yeah, it's not working.
No, here.
But
it's working.
Yeah, now it's working.
Okay.
So in this session of the in vitro models, I will shortly discuss the part of the work that we did in the task 1 of the bovine package 1, which was the generation and the characterization of the bovine cell lines. So I will specifically focus on the isolation and characterization of the bovine luteal cells that we performed in FBN in Dumastorf. So for the isolation of the luteal cells, we isolated the primary luteal cells from the corpus luteum, and then the cells were, the pure population of the luteal cells was separated by the Perkov gradient method. So, and further, the cells were cultured in a growth media, and also these cells were further cryopreserved for the later analysis. So as we know that initially the aim was to generate the novel immortal luteal cell lines. So for that, we cultured the primary luteal cells and transfected them with the plasmid vector containing the HTRT gene. And further, the transfected cells, they were selected in an antibiotic selection media, and the population, the cells that were transfected were further expanded and also subsequently passaged. So what we observed that post-passage 8, the cells they didn't proliferate, and though they carried the HTRT gene, but they failed to proliferate further. However, we were also running the non-transfected luteal cells, and we were quite lucky to proliferate them to a higher passage. And at this point, I characterized the primary luteal cells and also the luteal cell as the intermediate passage, that is the P15, and also at a higher passage, that is the P30. And so we wanted to look if the passage number has any effect on the functionality of these luteal cells. So as you can see, that the morphology of the higher passage luteal cells was significantly different because they become more enlarged and flattened as compared to the primary normal luteal cell morphology. And also the lipid content. The lipid content in the luteal cells is of utmost important because it is the substrate for the steroidogenesis, the synthesis of the progesterone, which is the main function of the luteal cells. So we see that the luteal cells, the primary luteal cells, they were quite heavily loaded with the lipids, but the cells at the higher passages, both at the intermediate and the very high passage, they reduced the lipid content. So that means that the functionality, the process of the steroidogenesis was severely hampered as we go higher with the passage. So next, we applied a linear model to look if there is an association between the cell doubling and the passage number. The data show there is a moderate association, a positive association between the cell doubling rate and the passage number. So the cell doubling rate, it significantly increases as the passage number increases. And also the concentration of the progesterone hormone was analyzed in the conditioned media from every passage, and we see that after passage passage 1, and we see at the passage 4, we see there is a significant reduction in the level of the progesterone, and it goes very, and at the very high, almost negligible amount of progesterone was observed. So next, we analyzed the expression of the key genes, the main functional genes which are important for the process of the steroidogenesis in luteal cells, and we see that the expression of the star HSDB, they were significantly reduced with the higher passage, and the LHCGR, the receptor at the luteal cells, we see at the 15 and 30 we didn't find any expressions. That means the LHCGR protein at the protein level also it was almost diminished. And similarly, we also look for the protein estimation of the star, which is the it's like a key rate limiting, we can say a rate limiting protein in the synthesis of the progesterone, and we see there is no traces of the star at the P15 and as well as the P30. And next, the passage number also hampered the cell proliferation. We see that the percentage of the luteal cell, the primary luteal cell, they have higher. They were higher in the S phase, but the proportion went down as we passaged the cells. And also with the passaging, the proliferation was delayed because if you see in the G2M phase, we have a high proportion of the G2M phase, which means the cells were in proliferation, like they were in the growth stage, but they have a very delayed proliferation, as we saw in the cell doubling rate also. The cell doubling going very high with the passage number. Next, I looked for the viability of these luteal cells, and what we found that the viability remains consistent irrespective of the passage number. So there was no effect on the viability, and there were about 90% of the cells each in primary luteal cells, and with P15 and P30 where the proportion was almost similar. So there was no effect on the viability. So this means that it could be that the several survival pathways, they are still activated in the luteal cells, and they remain uniform irrespective of the passage number. Next, we look for the structural integrity. So we stained the main cytoskeleton proteins, which is the vimentin and cytokeratin, and we found that these both proteins were co-expressed, and they were also consistently expressed irrespective of the lower or the higher passage number. And we also performed RNA sequencing on the early luteal cells. So this data comes from the P3, which is very high, very early stage of the luteal cells. And we found that there were approximately 13,763 genes that were expressed. And about topmost 500 genes, they were quite attributing to the function of the estrous cycle. So that means that these cells at the P3 were still functionally active. So in the summary, we can say that the in vitro passaging has adverse effect, at least on the signature molecules of the luteal cells. And however, but few functions like the structural integrity, the cell viability, these functions are retained, and they do not get altered even if the passage number is increased. And the repeatedly passage to proliferate, the key functions of the specialized cells, such as luteal cells, which are highly specialized cells, so they either diminish or they are altered. And these variations can potentially affect the experimental outcomes, also in the cell lines. And so it is very highly recommended to always cite the passage number, and also while using the cells in the in vitro models, especially in the cell line. So it is always important to always validate the key findings from the cell lines to their parental tissue, the primary cells. And also these results should always be included in the study, because we see in the luteal cells with the passaging, there's a complete functional breakdown of the functions in the luteal cells. That's it. Thank you very much for your attention.
So thank you, Arpna. So we have plenty of time for questions. Hiroaki? The microphone? Doesn't work? No? No?
Oh, yeah.
Okay.
So thank you very much for your talk, and I'm quite interested in what you established. And I was just wondering, did you check the responsiveness of the cell to RH? The first question, and the second question is, did you try to do any transfection of plasmid or siRNA and to see if the cells are okay to be transfected? Thank you.
The answer to your first question, I think we supplemented the growth media with the LH, luteinizing hormone, but with the later passages, the cells, they did not stimulate to the LH, because that's why we see also the LHCGR was a complete loss. So at the P15 and P30. So I think there is, with the passaging, there could be a desensitization of the receptors, which is a known phenomena. And to the second question, I think, no, we didn't do further. We just stopped there when we didn't see the cells were not proliferating when we transfected the luteal cells. So we stopped there. But interestingly, they controlled the non-transfected cells. They proliferated, and so we just analyzed that data.
Thank you very much. It is very interesting because I have never seen the luteal cell line like you established. So I think it's going to be very useful material for the research.
Yeah.
Thank you.
Yeah.
So further questions? Online?